In this video, we are going to discuss the antigen presenting cells. So in an adaptive acquired immune response, antigens have to be processed and presented to T cells or T lymphocytes in order for those cells to be activated. This is accomplished by those antigen presenting cells known as APCs. Recognize that the process is going to be dependent upon the type of antigen. Is it going to be an external, exogenous antigen, or is it going to be internal or endogenous? So let's look first at the exogenous or external antigens. The antigen is going to be internalized by the cells through a process known as endocytosis, and this is going to be a process that we did in phagocytosis video as well. So kind of look back to that for more details. But in an exogenous extracellular antigen, then what happens here is the antigen is going to be internalized again through endocytosis. This is going to, then you're going to have that phagosome, which is going to fuse with the lysosome, and then that leads to the destruction of the antigen. Once the antigen is digested into smaller pieces, it's then going to fuse with a vesicle containing that self-marker of the known as the MHC class II molecule, and then in order for it to be displayed, the antigen is then going to be loaded onto the MHC class II molecule. So you'll see here you've got the fusion of the MHC2 class II with the phagolysosome with those little antigen pieces, and here we have loaded the antigen onto the MHC class II molecule. Now the MHC antigen piece is going to be moved to the surface. So notice that the molecule moves, the vesicle moves to the surface, and now that antigen piece is displayed on the surface for presentation. So the MHC class II antigen complex is ready to activate, in this case, the T helper cells. Remember, only the macrophage, dendritic cells, and B cells use MHC class II to display antigens. So this is for MHC class II. Now, if we talk about an endogenous process, in the endogenous or internal antigens such as cancer or viral attacks, the antigen originates within the cell. Therefore, the cell is going to have to break down these polypeptide fragments, which are going to be cytosolic proteins, if you will, into an appropriate size for the cell to be able to utilize it. So here you'll see the antigen is an intracellular cytosolic protein. We're going to have to use a proteasome to break down that protein into peptides. So then you'll notice that they start from the inside, so that you're going to start with these peptide pieces. Okay, so here we have our peptide pieces that are intracellular. They are going to be transported to the ER, and in the ER the cell is going to load a piece of that peptide onto an MHC class I molecule for presentation. Ultimately, that MHC peptide piece is then going to have to be displayed on the surface, so you'll notice it gets moved to the surface, and then it is then located here, MHC class I, with a piece of peptide for presentation. In the case of MHC class I, all nucleated cells are able to process endogenous antigens in this manner. So all nucleated cells are able to act as an APC using the MHC class I for antigen display, because this is going to be an intracellular or an endogenous piece of antigen. So this is talking about APCs. Refer to the other videos on how the T cells then get activated in order to now destroy that antigen.